Got. Thank you. No, I haven't done anything. Let me see. Okay, great. Thank you. Um, Thank you very much, the organizing committee, for this opportunity and a wonderful lunch, actually. And thank you all for being here. My talk is about DNA-based vaccine technology. And we all know that the mRNA vaccine became a household name during the pandemic and was able to control the pandemic really successfully. But we were also able to identify limitations in mRNA vaccines. For example, as Ms. Regina Au talked about some of those challenges and limitations, one was the short duration of immune response. The immune response will wane down 3-4 months after the first vaccination. So multiple shots were given. Then also she talked about poor stability. mRNA vaccines require storage at frozen condition, -80, as Ms. Au talked about earlier in her talk before lunch. Then also the conventional vaccines protein, they have their own challenges. The manufacturing process is so complicated, it takes a long time. That's why mRNA beat protein in that sense. And then also the protein vaccines are not very effective against destroying the infected cell. That is, the T cell responses are weak. So DNA vaccines are well suited to overcome these limitations of the current vaccines. DNA vaccines have certain advantages in that sense, and that's target product profile. I think she talked about earlier very eloquently. So the DNA vaccines provide longer duration of antigen expression, which means that they could translate into longer durability of protection. Strong T cell responses means it elicits white blood cells that can go out and kill those cells that are now infected by the virus. Stability, um, normal refrigerator temperature, even at room temperature for manipulation after it's brought for vaccination. Manufacturing is flexible. So those are the target product profile for DNA-based vaccine. But one of the challenges with the DNA vaccine is the delivery. The delivery of DNA as opposed to mRNA is more challenging. And people use more harsher systems like electroporation, electric current. So you inject the DNA vaccine, apply a current that will take the DNA into the cell. Devices or viruses like J&J vaccine. So our company is developing DNA vaccine technology. We call it Placine. And that does not require a virus. So it's non-viral. It does not require a device. And it does not have lipid nanoparticles that some of the safety concerns like myocarditis, that's due to antigen, but also some safety concerns are attributed to the lipid nanoparticle. So we are using a conventional syringe and needle injection as opposed to devices or viruses. And we use a delivery system, a functionalized polymer that's shown here. And this polymer addresses the challenges in DNA delivery. As I told you, the DNA delivery is the key. It causes the DNA to be more bioavailable at the site of injection, disperse it through the tissue, protects against degradation, membrane interaction, also has adjuvant effect. Adjuvant means, as most of you know, it attracts the immune cells that build immune response from a vaccine. This shows that if you take a DNA vaccine in a test tube and expose to the enzyme DNase, it degrades the DNA. You can see the left several lanes. DNA was incubated with DNAs, and you don't see anything was chopped up by DNAs. But with our delivery system, you see the DNA bands are still there. So that's a protection of DNA against degradation by nucleases that are normally present in our tissue. And this shows that the gene expression is about 10 to 15 fold higher than if you just use naked DNA or unformulated DNA. Plascine is naked DNA formulated with our synthetic delivery system. I showed you that causes DNA protection, dispersion, stuff like that. So compared to naked DNA, you see 10-fold enhancement in gene transfer. So now I will talk about some of the limitations that are associated with current vaccine and how DNA vaccine addresses that, or the data. This shows, as I said to you earlier, that there's longer duration of antigen expression with DNA, the green one, as opposed to mRNA. You can see that within 2 days, there's a significant reduction in the expression of the antigen if you're using mRNA, and but as opposed to DNA. So, as I was saying, that mRNA immune responses wane down, quickly 3-4 months, and maybe because of this decline in the expression of the antigen, with DNA you can get longer expression durability. Okay. Then the T cell responses, the immune response. One type of immune response is a T cell response. You can see that after 12 months of vaccination, there's still significant T cell responses with our vaccines as opposed to mRNA. So that durability you're talking about, in fact, these are animal studies, translate into durability of the immune response, at least in animals. Another thing I think, Regina, I would mention, Ms. O, that dosing multiple times, that has been an issue. So um, here we are comparing a single injection of mRNA vaccine to our vaccine. And you can see that very quickly the mRNA is plateaued, doesn't change over time, doesn't increase over time, but the plascine, it builds up over time. So a single vaccine could give you longer range or durability of immune response. As you can see, the IgG responses in this animal study, and also the T cell responses are also higher with our vaccine. As I was telling you earlier, that since delivery is a challenging for DNA, then people have used electroporation devices, or a device, an Indian company, Zydus Cadeas, they have a needle-free device, not electroporation, but needle-free device with a pressure, it ejects. So here we are comparing with an electroporation device, and this is a complicated graph, but just pay attention to the orange color. This is the levels of antibodies with our vaccine. This is with electroporation. Earlier days, day 21, day 35, our vaccine electroporation, and this is the antibody response. These are the T cell responses. Again, our vaccine and electroporation, our vaccine electroporation. What I'm saying is that for mass immunization, if you have to give an option to take electrical current as a delivery system versus just simple needle injection, of course, compliance, something she talked about earlier. It was nice that her talk was before lunch, before this talk. So a lot of things I can refer to what Regina talked about. Then this is compared to viral vector. J&J is viral vector vaccine where you give single injection, because you can't give second injection. Adenovirus is common virus we all have. So second injection have antibodies against the virus. It won't work, even though J&J has some data that their second injection did work also. Here we're looking at comparing the antibody response is comparable to adenoviral vaccine side by side and conventional protein vaccine against SARS-CoV-2, but the T cell responses are lower, significantly lower in case of protein. So those are, again, you're trying to fill the gap, the challenges that you have seen in the current vaccines. Manufacturing, another thing she mentioned, that mRNA manufacturing is quite complex, and of course people don't know, and even though it's around for a long time. But DNA manufacturing is very generic. It doesn't matter what vaccine you're making, it's a very simple process. This is the vector that contains this antigen. It could be SARS-CoV-2, strain Delta, Omicron, whatever. Could be another pathogen. You simply take this out, put the DNA sequence of any pathogen or strain you want. The rest of the process is the same. That's not the case with protein. And also, you can put multiple antigens into one. For mRNA, you have to make each mRNA vaccine separately and then mix it. But here, one vaccine DNA vector can contain multiple antigens. So again, we're thinking about from a new generation type of vaccine, the things I've showed you, some of the challenges that it can overcome. Stability was the thing that I was talking to one of our attendee here earlier at lunch, and she used to give vaccination in Nevada, COVID vaccine, and you have to keep Pfizer at minus 80 degree. Moderna also frozen in the fridge. You cannot keep more than a few days, which is the 4 degree. Here we've shown that the stability of the vaccine at 4 degree of refrigerator for 12 months at least. This is every time you compare for immunization, looking at the antibody titer, neutralizing antibody, you do a freshly prepared, and you can see they're always comparable up to 12 months. So imagine keeping it 4 degrees centigrade. That helps a lot. I mean, that's why the mRNA vaccines are mostly used in USA and Europe, developed countries. But how about the third world countries where certainly having these freezers is a luxury? We've also looked at 37 degree, and we have seen stability for 2 weeks. Of course, you would like to probably want to show at harsher temperature. I think she mentioned Arizona or Nevada, maybe African countries. So again, storage wise, transportation wise, there is an advantage. That's a real advantage in terms of commercial viability of vaccine. So now, with those advantages of our platform, Plastine, I'll show you we're developing a new generation COVID vaccine. We call it Immunon 101. And here, this is a most recent variant, XBB 1.5. That's the FDA recommended in June. And that's the new vaccine that came out from Pfizer, Moderna, that has the most recent variant. Although that variant is also kind of gone over time, but that's what the current vaccine has. So we wanted to put that variant in our vector, and this is our delivery system with the adjuvant. We have demonstrated proof of concept with this new vaccine going to the clinic, and we plan to go into the clinic April of 2024 and rapidly advance into a phase 2 study in July in healthy volunteers. Preclinical proof of concept wise, this shows the IgG responses with our vaccine, primary injection and the boost, and then neutralizing antibody response that neutralizes the virus in an in vitro assay, the competition assay, and then the T cell responses. I always talk about, antibody response and T cell response. Antibody responses protect the virus from infection, clears it, but once the cell is infected, the T cells get rid of those infected cells. So T cell response is important to clear the infection completely. Sometimes you have vaccine people get that breakthrough you talked about, then still had some sickness, and that has to do with the not having clearing the infected cell properly. These are more immunology. I don't know if you guys are more involved in this. This is CD4 and CD8 cell. These are the actual T cells. All the data in the beginning, I showed you general T cell response, but now this shows what type of T cell. So CD4, CD8 cells are involved in the clearance, and we see those responses with our vaccine in mouse model. Now, before this clinical variant XBB 1.5, we did our vaccine with Delta, Alpha, and we have seen shown clearance in infected mice, challenged with the vaccinated mice were challenged with the virus. And then you see, complete clearance in this case and 95% clearance in this case in the log scale. So it shows that not only you see antibodies, neutralizing antibody, but also it can clear the virus upon challenge. Of course, you want to take from mouse to non-human primates. That's pretty standard before you go into humans. So we did a monkey study also, side-by-side comparison of the commercial mRNA vaccine. You can see The bottom means the virus is not there, clear. This is the clear stuff, the challenge. You can see that our vaccines, different placine vaccines, Delta or the Alpha variant, you can see the clearance is very comparable to mRNA. So now, going back to what you had said earlier, efficacy-wise, if you can show comparable efficacy, then what, But you have to have something more distinguishing. So as I told you, 4 degree, the temperature stability, durability, those are the TPP in terms of what's out there in the market. But these are all animal data. So if it all pans out in human being, that would be wonderful. But these are preclinical studies or generally directional. Okay, that means you should go into the clinic. This is the study I was telling you about. April of next year, we'll do a dose escalation of placine vaccine, and then it's a 1 month follow up for safety and immunogenicity. And you pick a dose where you have the safest dose and the most immune responses, then go into the phase 2 and add 75 more subjects to get a small study, not like Moderna, Pfizer, 40,000, 30,000 subjects. But this is the first vaccine. If you see some responses, then we can expand the study into a more registrational study. So the data we have, I shared with you, was with SARS-CoV-2, but we have looked at this placine approach. What is placine approach? Put the antigen DNA into a plasmid DNA. That's getting the recipe. And then a formulation that promotes the DNA uptake, protects the DNA. That's called placine. Of course, SARS-CoV-2, we also have data with other pathogens, flu, Lassa, Marburg, and monkeypox. This is some in collaboration with NID and IH on Lassa and Wistar Institute on flu and Marburg. I've just put one slide composite here to save time here. This is Marburg. You see IgG responses, T-cell responses with our vaccine. Same thing with influenza, IgG T-cell. Here Lassa, the T-cell response is really important. We don't have the IgG data yet, but the bottom line is that the immunogenicity you saw with SARS-CoV-2 is not just limited to that. You can expand into other pathogens. Then 2.5 minutes left. So we're also using the same approach for cancer vaccine. Now, in pathogen, your target is the antigen, spike antigen that you've talked about, or with Lassa or influenza, different type of antigen. Cancer is the same thing. If you can target the antigen of a cancer, either overexpressed in cancer or doesn't express in normal tissue at all, just in tumor tissue. If you can develop vaccine against those antigens, then you have a cancer vaccine, cancer therapeutic. So the company has an immunotherapy program where we have a immunotherapy completed just phase 2 randomized trial in ovarian cancer. But now this cancer vaccine application, Fixaplast, is that those antigens that are overexpressed in cancer tissue, but still expressed in normal also. But the Indiplast is individual, the personalized cancer vaccine, where a tumor in person X may be different than same ovarian tumor in person Y. Tumors are very different. So you target a specific antigen for a specific individual. So those are very early projects in our company. I'll show you the cancer vaccine data. This basically shows that if you take an antigen for a tumor and put into a placine, challenge the animal with the tumor, and then give your vaccine and look at the tumor profile. So we see a delayed in tumor growth in vaccinated animals and also extension of the survival. These are early experiments. We'd like to build this program over time. Again, with this cancer vaccine, also the T-cell responses are very important. We see that CD4, CD8 responses for clearance of the tumor. So with that, I would like to conclude that DNA vaccines, although there's no approved human DNA vaccine, there are a couple in animals, but this is probably has potential to replace or substitute the mRNA vaccine in terms of some of the challenges that I've talked to you about. So durability of the immune response with DNA. I showed you some data in animals. Also, you can put multiple antigens. I didn't show you the data, but we do have those vaccines as well. Stability at working temperatures, flexible manufacturing, independence from virus or device, which means better safety and compliance. You use the word compliance also. And so that has, I mean, I don't want to take a device or current versus, even the needle also hurts, but that's more conventional. So the compliance is better. So we do have preclinical proof of concept in non-human primates and mice, and going into human trial in April 2024. That will be the key. You can have a lot of preclinical data, but if it doesn't work in human, there's no point. And then, of course, our cancer vaccines also in a very early stage of development. With that, I'll stop and take any questions.